Okay, are there any questions? There's the worksheet. I guess that's it. Why was it so big? All right, my computer's going to be a little slow because it's still making the lecture file. Let me share my screen here. So we're only 89% done on the lecture file. It's still a little big, but I'll use that. That's too big. Out there. Well, that one's good. All right, today is June 22nd. No, July 22nd. And we are going to be talking about Lab 4 stains. Any questions about what we're going to do? Please remember you have a quiz 1, which must be taken this Wednesday. All right. Lab 4, staining techniques. We're going to talk about simple, differential, and special stains. There is some reading to do in the textbook. You should read about the size and the shape and the growth arrangement of bacterial cells, which is only 2 pages, 73 to 75. You should read about the bacterial cell wall on pages 80 to 83. You should read about the simple stain on page 65. You should read about the differential stains, the Gram stain on page 65 to 66, and the acid-fast stain on page 66 to 67. And then there are a few special stains we'll talk about. There's this negative stain for staining capsules on page 67 and page 75 to 77. There's the endospore stain. Oh, good. The lecture file just finished. My computer will now speed up. The endospore stain on page 68 and page 92 to 94. And the flagella staining on also page 68 and then page 77 to 78. Any questions about the reading? There are some video clips in today's lab and a virtual lab. So upon doing this lab, your learning objectives are 1, be able to prepare a bacterial smear for staining. Remember, when you make a bacterial smear, it is not part of the staining procedure. It is something done before you begin the staining procedure. And then 2, understand the principles and procedures used when performing the simple stain, a differential stain, a special staining, and then a special stain. So understand the principles and procedures used when you're doing. Like a simple stain, all you're doing is you make a bacterial smear, that's before the staining, and then you just apply one stain, and that's the simple stain. Interpret, and then 3, interpret the results for a simple stain, a differential stain, a special stain, and then be able to define the following terms right here. Obviously, the simple stain, the differential stain, a special stain, Acanthophilus, bacillus, a spirillum. What's peptidoglycan? What are gram-positive cell, a gram-negative cell? What's an acid-fast cell? What is a primary stain? What's a mordant? What's a decolorizer? What's the counter stain? What's the cell capsule? What's an endospore? And what's the flagella? Any questions about any of those terms? All right. Bacteria are essentially transparent. Therefore, it's necessary to stain them before they can be observed under the microscope. When you stain a cell, you need to make a bacterial smear before beginning the staining procedure. To make a bacterial smear, you just put a drop of sterile water on a clean slide, and then add the bacteria to it. Usually add only a little bit of bacteria and then mix it around, spreading the slide over the drop over the slide. You can also add cells in a liquid media by just putting a drop of the liquid media on the slide, meaning putting medias and cells on, and then spreading it around the slide. And then you let that air dry, and then you gently fix the organisms to the slide by gentle heating. We call that heat fixing, allowing the cells to attach to the glass so they're not washed off from the staining procedure, which will then later follow. A simple stain uses one dye. You note how I've crossed out the word basic. Doesn't matter which dye you use, basic dye or an acidic dye, it's still a simple stain. It allows you to see the bacterial cell, their shape, and growth arrangement. But you cannot distinguish differences between two different types of cells because all the cells will stain the same. A differential staining technique allows you to separate two or more types of cells when you're usually using two or more dyes. There are two simple stains that we're going to talk about, the Gram stain and the acid-fast stain. And then lastly, there's a special stain which is used to dye a particular bacteria structure, such as the cell capsule, the cell endospores, or the cell flagella. Any questions about any of that? All right, here we're seeing some cellular growth arrangements for cocci. You can see diplococci right there and there, streptococci right there, tetrads there, and then staphylococci right here. A few of the growth arrangements for cocci is not shown. What is one arrangement for cocci which is not shown? Anyone? Well, there's one very simple one, and that is a singlet. The singlet is not shown, at least here, and that's a single cocci. And then we have some growth arrangements for, actually, they're not growth arrangements for bacilli. These are different shapes. There's the spirilla. This is a pleomorphic cell. They're not very pleomorphic. Usually pleomorphic cells take on many different shapes. And these are different, but they're all, I don't know, similar. We're just going to word it that way. That's bacilli. And then there's a coccobacilli. Any questions about any of them? All right, let's talk about the differential stains. The first is the Gram stain, invented by Hans Christian Gram, and he had a name that we name it after, and so the Gram stain should be a capital G because it's named after Dr. Gram. I didn't mention it, but he made a differential stain where the patient's stains stain Gram negative, so their cells stain Gram negative, and the bacterial infection stain Gram positive. Obviously, that only worked when the infection was a Gram positive bacteria, but that's why he developed the stain, to differentiate between the bacteria and the patient's own cells. Microbiologists soon began using the Gram stain, and they discovered that it was differential among bacteria as well as archaebacteria, allowing people to distinguish the Gram-positive bacteria, which appear purple, and different from the Gram-negative cells, which appear pink. Any question about any of that? This looks like it's a little bit out of shape there. Maybe if I shrink it down, it'll be better. Well, it's still a little out of shape, but there it's good. Let's blow that up so I can see it better. There's that gram-negative bacillus, a gram-negative spiral. That looks like a gram-negative cocci right there, or coccus. And there's a gram-positive bacillus. There is a gram-positive coccus. Any question about any of that? The Gram reaction is based upon the cell wall composition of the bacteria. The gram-positive cell is different than the gram-negative cell. The gram-positive cell has a very thick peptidoglycan layer. Let me blow that way up. And there you can sort of see some of the layers. There's 1, 2, 3, 4. Can't see them there, but there's a couple in there, and then a couple here. And now this is too blurry. I can't see it. Let me shrink it down, and then maybe I can see. There's a layer there, several layers in here. But up there, I could see the layers. The point is, it's a very thick layer upon layer of peptidoglycan, and that's the main molecule in the cell wall of the gram-positive bacteria. For the gram-negative bacteria, the cell wall differs. Peptidoglycan is the main molecule in the cell wall, but there are other molecules. And there you can see there's several layers here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, at least 11 different layers of peptidoglycan in this gram-negative, but it's much thinner than the gram-positive bacteria. Also in the gram-negative bacteria, we have this lipid bilayer, which is called the outer membrane of the cell wall. And the lipid bilayer is similar to the cell membrane shown here. It is a little different in the outer layer of the lipid bilayer, and also has the saccharides attached to it here. Any question about any of that? For some reason, I lost most people. Did I lose everyone? No. So this is the outer membrane of the cell wall of a gram-negative cell, and then that's the peptidoglycan layer of the cell wall. After the bacteria have been stained with Graham's crystal violet, the iodine is then used as a mordant, which chemically complexes with the crystal violet to make a larger complex. Next, we use the decolorizer, which is applied just briefly to remove the crystal violet iodine complex out of the gram-negative cell, but not out of the gram-positive cell. If you do apply the decolorizer too long, you will pull it out of the gram-positive cells. And then if you don't apply it long enough, you will not pull the crystal violet iodine complex out of the gram-negative cell. So it is tricky applying the decolorizer, but if you do it correctly, you will only pull it out of the gram-negative cell and not out of the gram-positive cells, simply because of the thinness of the gram-negative layer compared to the thickness of the gram-positive layer of peptidoglycan. After the colorizing step, the gram-negative cells will be colorless, and the gram-positive cells will stain a purple color. You then apply the counter stain, which is safranin, and it will stain the gram-negative cells pink. Any question about any of that? There are some cells which are sometimes called gram variable. The Mycobacterium are the main family of bacteria, which are called gram variable. They have so much macolic acid in their cell wall, it's another molecule found mainly in the Mycobacterium, that it does not allow the gram stain to get in and stain the cells at least well. Usually the Mycobacterium are called gram positive because they stain with the crystal violet iodine complex and it's retained by the cell wall, but they do not stain as well as other gram positive bacteria. And sometimes they will be staining pink, even though some cells are staining purple. Like right here, this has been stained with a gram stain, and it's a Mycobacterium, so it's one species, but some of them are staining gram positive, and some of them are staining gram negative. What do you notice that's different between this cell here and that cell and that cell? And what's different between that cell and this cell and that cell? Anyone see a difference between that cell and these two? Or between this cell and these two? Nobody notices a difference?
The color?
Well, yeah, the color. What beside that?
How they're stuck together and how.
Yeah, the shapes, I guess.
There is a little difference in the shape. Let's go here, blow it up even further. This cell looks kind of ghostly. There's a little bit of a cell there, and then that part of the cell looks like a ghost. And this cell here, besides looking slightly ghostly, it looks like it's broken right there. And you also notice that both of these do not have a very distinct boundary between the cell and the outside of the cell. Whereas these cells have a very distinct boundary between the inside and the outside of the cell. And that's because these cells are viable. The cell membrane and the cell wall are intact. There's a very distinct boundary. That cell is clearly broken, and that cell wall is broken, and so that's a dead cell. And this one looks ghostly because it too is a dead cell. And what happened is somebody did a gram stain on an old culture of Mycobacterium, and then all the dead cells are staining gram negative, and then all the viable cells are staining gram positive. And that's why.
So all gram cells that are negative are dead cells.
Well, all the ones that are staining gram negative are probably dead or dying cells. There might be some that are dying, so they're all the dead ones are probably gram negative, yes. Okay. But some of them may be dying cells. Like that one there looks pretty good. It might be dying. But the point is that the gram stain is supposed to be done on cells that are 2 days old or less. And I'll bet you that whoever made this slide preparation did it on cells that were at least a week old, probably 2 weeks old. And every time I've seen the Mycobacterium that students have stained and they got this results, I asked them, how old were these cells when you stained them? And everyone told me that the cells were at least 2 weeks old, in which case they had a lot of dead cells in there, as well as a few viable cells. And the viable cells stain gram positive, the non-viable gram negative, and maybe some of the dying cells are also staining gram negative. But this is the only time I've ever seen the gram variable results. When students are staining Mycobacterium and the cells are too old to do the gram stain. Okay, It's the only time I've ever seen the gram variable results. If ever the student used 2-day-old Mycobacterium or less, then the cells always stain gram positive. Now, admittedly, I've only stained 2 or 3 species of Mycobacterium, but in all the ones I've done, actually, I always got the cells to always stain gram positive because I never stained old cells. And then when I looked at students' cells, if ever they got gram variable results, it was always the family Mycobacteria, and it was always cells 2 weeks old or older, which they should not do a gram stain on cells that old, because the gram stain may not stain correctly if the cells are dead, and that cell is definitely dead. You can see the break right there. So to prevent gram variable results, avoid using old cells. Prepare a thin smear. If you have a thick smear, it's going to be trouble for the dye getting through the layers of cell to get to the bottom cells. And then when you're doing the decolorizing step, it's going to be hard for the decolorizer to pull out the crystal violet iodine complex from the bottom layer of a thick layer of cells. So always make thin smears. Heat fix the bacteria on the slide only after the smear has air dried. If you perform it before the water has evaporated, then you'll get the water to boil, which will put holes in the cell membrane and the cell wall, which will obviously interfere with the staining. And then avoid overheating the cells during heat fixation. If you overheat the cells, that will disrupt the cell walls. It will actually make the cells appear to look melted. So you have a nice, let's say you have a nice square shape. The cells are going to be melted and the boundary won't be distinct and square. It will be rounded. Obviously, most bacteria, except for Haloarcula, are not square in shape. I'm just giving that as an example. All right, any questions about the gram stain? All right, some bacteria stain acid fast, namely the family Mycobacteria and Nocardia. The reason why these two families are acid fast staining cells is because they have a waxy substance in the cell wall. It's called mycolic acid, and the bacteria may have a lot of mycolic acid in the cell wall, where the mycolic acid is actually about similar in number to the peptidoglycan in the cell wall, at least of the family Mycobacterium. There's a lot of mycolic acid in the family Mycobacterium. The mycolic acid keeps the cells, the bacteria, to resist the decolorization of the acid-fast stain. And these are referred to as acid-fast positive bacteria. So Mycobacterium are acid-fast positive bacteria. And then most other families, except for Nocardia, are acid-fast negative bacteria. The acid-fast stain will come out of the other cells in the decolorizing step. Initially, all the acid-fast and non-acid-fast stains take up the primary stain, which is Carbol fusion. It's a cherry red color. And then the cells are decolorized with acid alcohol, which pulls out the acid-fast stain from the acid-fast negative cells. It does not pull out the acid-fast stain from Mycobacteria and the Nocardia, which are acid-fast positive bacteria. You then counterstain the cells after they decolorize. The counterstain can be blue or green depending on which counterstain is applied. And then you look at the cells under the microscope. If you have pink or reddish cells, let me blow this up a little, then those are acid-fast positive cells, like there's one there. And then they're greenish or bluish cells, they're acid-fast negative cells. So this is probably from somebody's sputum, and acid-fast positive from maybe Mycobacterium tuberculosis. And then those would be other bacteria in the person's mouth or throat, which are acid-fast negative. All right, any question about the acid-fast stain? All right, let's turn on to the special stains. The first special stain we're going to talk about is the negative stain, which is used, at least in this case, to stain a capsule. You can also call this a capsule stain. Some bacteria produce a gelatinous substance called the glycocalyx, which has polysaccharides and other molecules in it. It's secreted exterior to the cell wall. If the glycocalyx has other molecules in it, meaning other than mostly just water, and it's attached firmly to the cell wall, and you can see it with the light microscope, we call it a capsule. If, on the other hand, the glycocalyx is mostly water, you cannot see it with the light microscope. It is called the slime layer. The capsule can be associated with an organism's virulence, as the capsule protects the cell from being phagocytized by white blood cells. If the cell produces a capsule, they can often look a little different when grown as a colony, and these are mucoid colonies of Streptococcus pneumoniae, and they're mucoid because the cells are producing the capsule. Capsules are not easily observed with conventional staining methods. Usually with the Gram stain, the cell capsule is washed away and does not show with the Gram stain. Therefore, we use a gentle procedure such as the negative stain to view the capsule. Where India ink is used in this position, this picture, or in this one, nigrosin, and perhaps another stain, meaning two stains might be used here to stain this, because that color looks a little different than that one. So I'm guessing two stains are used here. If the capsule is present, the capsule will not stain and will appear to be a clear region around the cell. India ink stains the background very darkly, and then it tends to stain the cell lighter, and that's why it's kind of a different shade of gray. Anyways, both of them clearly show the capsule, where a clear region, non-staining, is shown in the negative stain, also called the capsule stain. All right, another special stain is the endospore stain, which is used to show endospores, a special structure made in certain species of cells to allow the cell to survive harsh environmental conditions. Endospores are metabolically inactive, hardy structures. They're a defensive strategy that some species make in order to survive harsh environmental conditions. So when the environment starts decaying and becoming less favorable for the parent cell to grow, the parent cell if it is a member of two families, mainly Clostridium and Bacillus, they can make an endospore inside the parent cell. And when that endospore is finished, it can survive harsh environmental conditions where the parent cell will die. Once the environment then becomes favorable, the endospore can germinate and return to the normal metabolically active vegetative cell that we used to call the parent cell. Obviously now it's the offspring cell, but it can then reproduce and grow. Any question about that? Most of the bacteria that produce endospores are in the genus or family Bacillus and Clostridium. There's both a genus called that and a family, and the genus is in the family, so we're just going to call the two the same, family and genera. There are one or two other family that also produce endospores, but most of them are in the Bacillus or Clostridium. And I think there's only one species in each of the other families that produce the endospore. Excuse me. Here is a table listing some pathogenic endospore forming bacteria. They're all bacilli. There's the family Bacillus, like Bacillus anthracis, causing the disease anthrax. Bacillus cereus, causing food poisoning. Clostridium botulinum, causing botulism, and obviously food poisoning causing the botulism. Clostridium tetani, causing tetanus or lockjaw, and a few other members as well. You'll note that the family Bacillus is aerobic, the family Clostridium is anaerobic. All right, any question about the endospore stain so far? If not, let's talk about the Schaefer-Fulton endospore stain. It is an endospore stain invented by Schaefer and Fulton. It uses malachite green as the primary stain, which initially dyes both the endospores and the vegetative parent cells green. Heat is then applied as a mordant to help the malachite green get inside the endospore. You then decolorize the cells just with water. Water will pull the malachite green out of the vegetative and parent cells. You then stain the cells with safranin. Safranin is the counter stain. Remember, a counter stain is just the second stain you use that is a different color than the primary stain. Let me blow this up a little. Here we see an endospore which has come outside of the parent cell. You'll note that it is Coccobacillus in shape. And there's the parent cell or the vegetative cell. And you'll notice that it's at a rod or a bacillus with rounded ends. Here we have the endospore growing within the parent cell. Let me blow that up even further. So right there we have the green endospore within this pinkish parent cell. So there's a little bit of the parent cell on that side and most of the parent cell on that side. Any question about endospores? Remember, endospores are just a survival structure. They allow the species to survive harsh environmental conditions. Another special stain is the flagella stain, which is a stain to show the flagella. If the cell can move, meaning it has motility or locomotion, and it's a bacteria, or even an archaebacteria, it can only move because it has one or more flagellum. The flagella are composed of the flagellin protein. You can classify the species by the number and distribution of flagella on the bacterial surface of the cells. There's Monotrichus. The species has only one flagellum. There's Lophotrichus. The species has multiple flagella on one side of the cell. That's why we call it polar. There's Amphitrichus, and that has flagella on both sides of the cell, called polar. And then there's Peritrichus. The cell has flagella all over the cell. Any question about any of that? Now, when you look at cells, if they have more than monotrichus, actually even monotrichus will have two types of cells. And if they're peritrichous, they will have all the different types of cells, meaning the monotrichous, some of the cells, when you look at them, they will be monotrichous, having one flagellum. But some of the cells will have no or zero flagellum. And the cells which are amphitrichous, some of the cells will be amphitrichous, some of the cells which are peritrichous, some of them will be peritrichous, having flagella all over the place. But some of them which are in with the peritrichous species will be amphitrichous, and some of them will be lophitrichous, and some of them will be monotrichous, and some of them will be having no flagellum on the bacteria, even though they're all the same species. And let's look at an example here. I don't want to go too high because then I can't see the flagellum. This is clearly peritrichous. So is that one and that one. But that one there looks like it only has flagella on one side of the cell. Same with this one here. Flagellum on one side of the cell. More than one. This one there only has one flagellum on one side of the cell. So there, that one there is monotrichous. The one with two or more is lophitrichous. Let me see if I can find a amphitrichous. That one there might be amphitrichous. It's hard to say because that one could be from this side. That one has several on it on this side. And it looks like that right there is on this side. It's not this one here. So that one looks like amphitrichous. And if you look real hard, that one there might have no flagella. When the cell first divides, it will have no flagella. And then the cell will put out one flagella, and then it'll put out multiple flagella on one side, where is that? There it is there. And then it'll put out multiple flagella on both sides, like this one here. And then it'll put flagella out everywhere. So that's why when you see a species which is peritrichous, you will see some amphitrichous, some lophotrichous, some monotrichous, and some with no flagella or no flagellum. Any question about any of that? So how do you name them? You name them by the highest order of flagella you see. So if the highest order you see is amphitrichous, you say that species is amphitrichous in its flagellar arrangement. Any question about any of that? You'll need to know this only for the lab. I'm not going to quiz you on this for a quiz. Flagella are too thin to be seen with the standard staining technique. So if you stain them with the gram stain, you will not see the flagella. Special stains are required to view them under the light microscope. A mordant is frequently used with a special flagella stain to help coat the flagella, and then you just put on multiple coatings of the mordant until there's so many coatings you can actually see the flagella. All right, any questions about the flagella stain? If not, here's a few terms you should know for this lab. You can skip the references. Now let's talk about the laboratory exercise. You're not physically going to be performing any of these activities, but you must understand how to prepare a bacterial smear for staining. You should know that a bacterial smear is something you do before you do any of the staining. Is that clear? So they're two separate procedures. One procedure is to stain the cell. The other procedure is to make the bacterial smear. And you must do the bacterial smear before you do the bacterial staining. But you don't ever have to do if you do the bacterial smear, you don't ever have to do the staining. And there are some stains, meaning some stain applications, that you do not make a bacterial smear before you stain the cell. Like when you make a wet mount and then you stain the wet mount, where you add the stain to the wet mount to colorize the cells, that is not making a bacterial smear, but it is making a stain. Any question about any of that? Anyways, you must know the various staining techniques and be able to interpret the results using the microscope. So answer the following. For procedure 1, we're preparing a smear for staining. View the following video clips on preparing a bacterial smear and heat fixing the slide. Before you heat fix it, remember you have to air dry. So you make a bacterial smear, air dry the slide, and then you heat fix it. The air drying and the heat fixing will attach the bacteria to the glass so the cells do not wash off. And this is the procedure for making a thin bacteria smear. You should only use a little bit of cells so you have a thin single layer of cells in your bacterial smear. And the first question for procedure 1 is number 3. What are 4 common errors when preparing a bacterial smear for a stain? So that's your first question. I've got them all with a question mark. Procedure 2 is looking at the simple stain. Here's a video clip for making a simple stain. Review the steps for performing a simple stain. First, you add a basic dye. After about a minute, you wash off the dye. Well, I guess first you start with a bacterial smear, and then you add the basic dye to it. And then you leave the dye on for 1 minute, wash it off with water, blot it dry with bibulous paper. Make sure you don't rub the slide. And here we see a video, not a video, pictures of a simple stain. There's a simple stain of E. coli and a simple stain of Staphylococcus aureus. Please note that the E. coli are sometimes hooked together. That right there is 2 cells. E. coli is not bent like that. And here's another example, 2 cells. And then that's probably 3 cells right there, stuck together. So for procedure 2, question 3A, describe the cellular morphology, that's the shape of E. coli. And then describe the shape of Staphylococcus aureus, shown here. And then what's the total magnification for these pictures here? Meaning when we took these pictures, what magnification, total magnification did we use? And it is stated in the procedure. If it doesn't give the total magnification, it tells you the objective lens used. And you know that we always use 10x ocular lens. So the total magnification is just 10 times the objective. All right, for procedure 3, we're looking at the Gram stain. View the clip of the Gram stain. It's a little video. And then practice the Gram stain using the virtual lab. Come on, open. This may take a little while to open. Boy, this is taking a long while. There it goes. Come on, come on. I have a fairly fast internet connection, but it wasn't downloading very fast now. So once you get here, click continue.
Hi, Lab Assistant. Welcome to the yogurt section of the dairy processing plant. It appears that our latest batch of yogurt might have been contaminated by some undesirable bacteria.
So you can read it or listen to it.
All yogurt has bacteria in it, since yogurt is made by adding specific bacteria to milk. These bacteria are beneficial to humans.
I'm going to skip ahead to where we
This
get
is
the
what
procedure.
Now take this yogurt. In the gram bacteria that's bacteria. We are going. All right, let's prepare our slides for gram staining. To prevent bacteria from contaminating our work area, we have wiped down the table with a 70% ethanol solution. We will also light a Bunsen burner to sterilize the inoculating loops we'll use later and help maintain a sterile working environment. You're going to use a striker to light the Bunsen burner.
So
The gas is turned on low already, so bring the striker to the Bunsen burner with the flint side facing down and squeeze the striker to create sparks.
what you do is you just take the mouse and drop this down, and it sparks itself.
We need to dilute the yogurt before we put it on the microscope. Start by placing 1 milliliter of the diluent into a test tube using a pipe. This is an inoculation. Start by sterilizing the inoculation loop in the flame of the Bunsen burner. After it has cooled down, place the yogurt sample into the previously Now,
I'm
use
skipping
the
ahead to the gram
inoculation.
stain.
Sterilize the inoculating loop again in the flame of the Bunsen burner. Hold it direct. Transfer a small amount. Place 2 loops full from the sample onto the slide.
So you just follow the arrow with your mouse.
Tilt the slide to spread the dropout. Now we'll heat. Place the slide in a clasp, such as a clothespin, and pass it over the flame of the Bunsen burner. Now we begin the staining process. After the staining is complete, we can look at these slides using a microscope.
By the way, that was not correct. It showed you a liquid culture of cells that they heat fixed. You never want to heat water on the slide. You want that slide to air dry before you heat fix the cells.
We use 4 chemicals to stain a slide. The first is crystal violet. This is our purple stain, which will indicate gram-positive bacteria. Using the dropper, squeeze out enough drops to cover your sample.
So what you do is you just go down,
Let that sit for 60 seconds.
let that go for 60 seconds. You have to continue to get the clock to go.
Now rinse it under slow running water. Now use the bottle of Gram's iodine. Gram's iodine will fix the dye to the cell walls of the bacteria and make it permanent.
Of the gram-positive cells, it'll make it permanent, assuming you don't over-decolorize.
Squeeze several drops onto the sample. Let that sit for 60 seconds.
Anyways, that's how you do it.
Now,
Any
rinse it
questions
under
about any
slow
of that?
running water.
All right. So do the gram stain, the virtual lab. I believe there's a question from the virtual lab in the lab, and you have to get that question right to get 100% on the lab worksheet. So view the virtual lab and do all of the gram stain. Here's the procedure of the gram stain: flooding the smear with crystal violet, then flood the smear with iodine, flood the smear with the decolorizer, let it soak for only 5 seconds. It's best to apply the decolorizer while you're holding the slide up at an angle next to your eye, and then watching the crystal violet run down the slide. Once the purple color stops running down the slide, stop adding the decolorizer. And that will be about 5 seconds. With the decolorizer we tend to use at Clark College. There are different decolorizers. In the virtual lab here, the decolorizer they are using is only ethanol. I think it's 95% ethanol. But the point is, it's only ethanol, and it's a little weaker. And so when they're applying the decolorizer, it's going to be longer than 5 seconds. It's something like 20 seconds they're applying it for, and that's because their decolorizer is weaker. And then after that, you wash the slide immediately to remove the decolorizer, and then flood the smear with safranin, letting it soak for 1 or 2 minutes. And then you wash that off and let the slide dry with bibulous paper, and then you can look at it under the microscope. And this has the same procedure I gave in the lecture with the crystal violet, all the cells stained blue-purple. With the addition of Gram's iodine, the cells will be a little darker staining, which I might even see there a little bit, maybe not. And then there'll be a little less blue-purple, a little more purple in color. And then you add the decolorizing, and after decolorization, the gram-negative cells will be clear, the gram-positive cells will be purple. And then you add the safranin as the counter stain, and then the gram-negative cells will be pinkish in color, the gram-positive cells will be purple in color. Any question about any of that? And then complete the table. So the gram reaction of E. coli, state whether it's gram-positive or gram-negative right here. And the cell morphology, state whether the cells are a coccus or a bacillus. For Staphylococcus aureus, the gram reaction stated here, and then state the cellular morphology here. And then here we have the cells stained with the gram stain. Let me shrink that down or blow that up. Something's not quite right there. This for some reason got moved. Hopefully yours didn't get moved. Can I move that? Kind of. Yeah, that's better. So this is the E. coli, and that's the Staphylococcus aureus. Any question about that? All right. For procedure 3, also do 3A, B, C, D, and E, and F. For Staphylococcus aureus, after the primary stain is added, state the color. It's either pink, purple, or colorless. And then E. coli, after the primary stain is added, give me the color right here. For Staphylococcus aureus, after the alcohol acetone step, give me the color there. For E. coli, after the alcohol acetone step, give me the color there. For Staphylococcus aureus, after the counter stain is added, give me the color here. And then for E. coli, after the counter stain is added, give me the color there. Some students overlook this and don't fill it in, and this will be worth 1.2 points. So you want this filled in. Any question about any of that? For procedure 4, we're looking at the acid-fast bacteria. View the following video clip for doing the acid-fast stain. And this is the steps when performing the modified Kenyon acid-fast bacteria stain. There is more than one acid-fast bacteria stain. This one we're only talking about the modified Kenyon acid-fast bacteria stain. First, you apply carbol fusion. It's the primary stain. It stains all the cells pinkish or reddish. You then apply the acid alcohol wash. That's the decolorizer. It will remove the carbol fusion from the acid-fast negative cells. The acid-fast positive cells will retain the carbol fusion. You then add brilliant green as the counter stain to stain the acid-fast negative cells a green color. And then the acid-fast positive cells retain the pinkish color. Any question about any of that? Below are two smears viewed at 1000x power that were acid-fast bacteria stain. Interpret whether the organisms denoted by an arrow are acid-fast positive or acid-fast bacteria negative. So that one, state here if it's acid-fast positive or acid-fast negative. If you can't type it there, or you don't want to type it there, like actually I can't get there, I'm getting up here, I'm trying to get there, then just state in the Zeal-Nielsen method, and for some reason this is off too. Hopefully yours won't be off. Hopefully that's right. Zeal-Nielsen method and the modified Kenyon-Cole method. Anyway, state whether the leftmost arrow in the first picture is acid-fast positive or acid-fast negative, and you can type it here or here. Just put in more space if you need it. Or you can type it there if you can. For some reason, I'm not able to get there. I guess I can type there, but not here. And then state whether that one is acid-fast positive bacteria or acid-fast negative. State whether that one's acid-fast positive or acid-fast negative. State whether that one's acid-fast positive or acid-fast negative. Another way to do it would be to say whether the cell, if it's green, state whether it's acid-fast positive or negative, and whether it's pinkish, if it's acid-fast positive or negative. But state it someplace to get your correct answer. And then go on to procedure 5. We're looking at special stains in procedure 5. We have a video clip on the endospore stain, so watch that. We have a video clip on the negative stain used for staining capsules, so watch that. And below we have 3 bacterial smears being observed at 1000x power. Figure 2A for procedure 5, 2A, right there goes to question 2A. This is the Schaefer-Fulton stain which uses wet stain as the primary stain, and it ultimately dyes what? Which are produced by some bacteria to aid in survival. Any question about any of that? You can find the answer in the lab module if you don't know. And then that's 2A1. So I guess that's 2A1, 2A2, 2A3 lists some examples of bacteria that are pathogenic endospore-forming bacteria. And you got that in the table in the lab module. Some examples is more than one species. And then for procedure 5, 2B, this is what type of stain that coats long whip-like appendages which aid in motility. So if the bacteria move, what type of stain are we using to show the structure that causes the cell to move? And then state the flagella arrangement type for the cell shown in 2B here. And then list some examples of clinically significant bacteria that produce flagella. You can find that in the lab module as well as online. And then for 2C, this is what type of stain that uses India ink or nigrosin and can be used to detect the presence of what cell structure, which is located exterior to the cell wall and can prevent phagocytosis. So that would be 2C1, 2C2. Remember when you're doing either C1 or B1 or A1, that you don't say it's a special stain, because all of them are special stains. I want to know what type of a special stain. Okay? And then list the structure of the cell in 2C2, procedure 5, number 2, C2, which is located exterior to the cell wall and prevents phagocytosis. And then for 2C3, list some pathogenic microorganisms that produce a capsule. And that you can find in the lab module. Any questions about any of those questions? In this lab, we also have 2 case studies. We have in case study 1, a 45-year-old female experiencing respiratory issues. Based on her clinical presentation shown here, the doctor orders an X-ray, and this X-ray is not normal. Normally, the lungs are very black like that, and this region shows something going on in her lungs. In addition to the chest X-rays, they take an acid-fast bacteria smear on a sputum, right there, the sputum smear, and then they do the Zeal-Nielsen stain. In this case study 1, you need to state the species of bacteria causing the infection. Don't just tell me the disease. You need to tell me the species causing the disease for case study 1. For case study 2, we have a 64-year-old diabetic patient admitted to the hospital with soft tissue necrosis, shown right there. A routine culture and gram stain is ordered. The bacteria grew only anaerobically. Based on the findings below, what is the most probable diagnosis? You must name the species causing the infection. So there's the patient's foot. There's the gram stain. So you can see if it's gram negative or not, as well as the shape of the bacteria. You also see the Schaefer-Fulton stain saying a structure in the cells. You have to figure out what structure that is, and then go look up what species caused this infection. You just need to look in the lab module. All right, any questions about the case studies? All right. This is week 3. We will go with what the students voted on, and I haven't looked yet for whether this lab will be graded 1 time or 2 times. Any questions? If not, go ahead and get started on the lab. I will be here at least till 8:30. I may be here as long as 9 o'clock if students need me. And I'm here to answer any questions concerning the lab or the course. And I guess I should state, I will be here until the last student logs out. So if you have a question, make sure you ask before the last student logs out. Okay, I just looked at the way the class voted, and actually everybody who voted wants there to be 2 gradings to the labs. So for this week's labs and all other labs in this term, you will have the labs graded 2 times. All right, bye.